So as Alice mentioned, my name is Bhatt, and I am an associate professor of medicine and of genetics at Stanford University. The title of my talk today is Precise Microbiome Genomics to Precision Medicine. While not particularly for the topics that I'll discuss today, I did want to share my disclosures with you first. So first and foremost, it's important that I acknowledge my really fantastic research group. And this is a picture of all of us, or rather, as you can tell, a collage of many of us at Cold Spring Harbor Microbiome meeting that happened in October of 2022. For those of you who are interested in the microbiome, we'll be having another such meeting at Cold Spring Harbor in October of 2024. And I encourage you all to check it out and consider attending. So in our lab, and indeed, I think at the cornerstone of every aspect of biology, we hope to measure what matters. No matter what type of scientist you are, be it a wet lab scientist, dry lab scientist, at the end of the day, what we do is we identify molecules of interest, macromolecules of interest, cells of interest, and then we measure them. And we measure them with the hope that we can relate those measurements to something meaningful from a biological perspective. Of course, is no exception to this rule. And in the microbiome sciences, we also seek to measure what matters. But I would actually challenge us a little bit in this notion, because while we seek to measure what matters, what we tend to do in most of the sciences, the microbiome included, is we measure what is convenient. We measure what we know how to measure. We measure what other people measured before us. And that means that we don't always get exactly the answer that we're looking for. And so over the course of this presentation, I'll be talking about three different topics where our lab has sought to not measure what is convenient, but measure what we think might biologically. In the first vignette, I will speak about the importance of looking at the microbiome from the standpoint of absolute abundance as opposed to relative abundance. I'll then speak of considering bacteria and eukaryotic viruses when we carry out microbiome profiling. And I'll speak about virome inclusive profiling methods that we've been working to develop. In the last section, I will focus on inversion, DNA flips within the genome, as an interesting mechanism of genetic variation that has to date been underappreciated in microbiomes. The first section is the work of Dylan McGeaney and Mai Dvorak. Mai Dvorak is a PhD student in the lab, and Dylan McGeaney is a very recent graduate of the lab, having just defended her PhD thesis about 2 weeks ago. A question that they asked was, what if things like cancer immunotherapy response aren't related to relative enrichment or depletion of certain bacteria, but rather might be related to the absolute abundance of bacteria that are present in a gut microbiome? Many of you may be familiar with the fact that in 2018, three very impressive papers came out in Science from the Gajewski, Zitvogel, and Wargo groups that demonstrate that microbiome composition, meaning which taxa are present and absent, was associated with whether or not cancer patients responded to immunotherapy. Now, while this result was very exciting, one of the things that was a little bit vexing and perplexing was the fact that these three papers that came out back to back to back all found different compositions to be associated with response. One of the things that we know in biology is that concentration matters. If you were to do a biochemical experiment, for example, you'd need to know what concentration you were adding your drug to a cell culture at, for example. Like to go to the physician and get blood work done, your blood work results would be reported as absolute concentrations, the number of cells per microliter, milligrams per deciliter of a compound, not the relative amount of to lymphocytes. So why then in space do we insist on focusing on relative abundance? Well, as with RNA-seq-like data, One of the reasons that we focus on relative abundance is because our methods, 16S sequencing and shotgun metagenomic sequencing, very well suited to giving us information in the relative abundance, but they can't provide us information, at least when carried out as is usually done, on absolute concentration. So, to summarize, what we do right now in the microbiome field is we take a stool sample when we're studying the gut microbiome, we extract DNA, you could also extract RNA, and then plot the relative abundance of organisms to one another, as is present in the plot on the right-hand side of this figure. Here we have 80% of the blue organism and 20% of the yellow organism. We then carry downstream analyses based on these relative abundances. But I would argue that most of the microbiome measurements that we perform in the field are flawed, or at least very limited. Take, for example, this toy example. Here, the top part of the graphic, we have an individual who is at day 0, who has 80% of the blue organism and 20% of the yellow. If you move over to the right, you can see by day 14, this individual is 50-50 blue and yellow. Now, of course, in the absolute abundance space, this could be explained by a variety of different findings. It could be that we started out with 8 blue organisms and 2 yellow organisms, as is pictured here on the bottom. And we could end up with 2 and 2, meaning we have loss of the blue organism, or we could have growth of the yellow organism and end up with 8 and 8, or anything in between. Obviously, the consequence of having 4 per unit of volume versus 16 organisms per unit of volume might have an impact on biology. So there are ways to quantify absolute concentration of bacteria, or sometimes referred to as microbial load. Here and moving forward, we're making the assumption that the stool sample is representative of the colon content. Methods that others have used include microscopy and counting, 16S ribosomal RNA fluorescence in situ hybridization, or FISH, and then flow cytometry, qPCR for universal marker genes, spike-ins, and then, of course, droplet digital PCR can be used. For the purpose of our efforts, we decided to focus on 16S quantitative PCR. All of these methods have been described before, but despite the fact that they exist, a very tiny proportion of microbiome studies actually report absolute concentration. I would venture to guess less than 1%, or perhaps even less than 0.1% of all microbiome studies report an absolute concentration. And so what we sought to do was to further develop methods that would be easily applied by labs like ours. Based on that, we figured that among these 5, the 16S quantitative PCR approach would be most easy for us to use in our setting. Why do this? Well, it's been demonstrated that absolute concentration of bacteria in the gut of animals can actually change with different biological circumstances. Take, for example, this manuscript that was published in 2015 by Clark et al. Here, what they did was quantify the absolute of different types of bacteria that were present in Drosophila guts over time. On the y-axis, you can see the normalized 16S abundance over a 10-day median. And on the you can see the age of the Drosophila in days. You have on the left gamma proteobacteria in the middle and alpha proteobacteria on the right. And as you can see, for every single type of bacterium that was evaluated in this context, the absolute concentration of bacteria in the Drosophila gut actually increases with age. And so there are biological phenomena like aging that actually are associated with increased bacterial concentration. Now, we sought to carry out this experiment not only to test and implement absolute abundance concentration, but also to figure out how common storage and processing efforts can impact the measurements that we make in microbiome studies. The reason that we had undertaken this was in most of our studies at Stanford, we collect samples from individuals who are at Stanford Hospital, which is only about 500 meters from where we work. It's a quick walk. We can walk over to the hospital, collect the sample, and bring them back to our lab and process them, freeze them right away. We can do this without adding any preservatives to our samples, and that works really fantastically well. But in collaboration with colleagues at the H3 Africa Genomics Consortium, including Voki Waran and Scott Hazelhurst, we had decided to undergo a process whereby we could collect samples from many different geographic locations within Africa, both rural and urban settings, where it would not be possible to immediately process and then freeze those samples. Because of that, we wanted to understand how commonly used preservatives, such as the OmniGut OMR DNA Genotech 200 and the Zymo DNA RNA Shield, would function in preserving taxa that are present within a sample. In this study, what we did was we took stool samples from 10 donors, and split them, and either preserved them without any preservatives, putting them at minus 80 degrees right away, very typical for what we would do in our routine lab work at Stanford. We also took some of the sample, put it in the OmniGene preservative, and some and put it in the Zymo preservative. For each of the preserved samples, we either put them immediately into the deep freezer. We kept them at about 23 degrees Celsius for days, or we put them at 40 degrees Celsius for 7 days to mimic the types of preservative storage temperatures that these samples might be experiencing in places like Burkina Faso and Ghana, where we were planning to collect samples. We then put all of these samples after 7 days into deep freezing. We then extracted DNA and did metagenomic sequencing and 16S quantitative PCR. We extracted RNA and did metatranscriptomic sequencing. Overall, we did all of these experiments in triplicate, and so you can imagine even from a simple 10 donor, we ended up with many, many, many experiments that we needed to evaluate. Now, this is a busy slide, but just to summarize, I'm going to take you through row by row. Let's start at the top row. The first thing that we need to do is we need to generate a standard curve of 16S using primers that are common to conserved regions of the 16S ribosomal RNA gene. We created standard curves for 2 different organisms, Bacteroides vulgaris or P. vulgaris and F. prausnitzii. Bacteroides vulgaris is, of course, from the Bacteroidetes phylum, and Faecalibacterium prausnitzii is from the Firmicutes. We created a dilution series, made this standard curve, and that enabled us to take these CQ values that we get from our quantitative PCR machine and relate them to absolute copy number of the 16S within a sample. And in the middle row, performed 16S ribosomal RNA quantitative PCR on our extracted DNA from our stool samples. We then related this information back to the dry weight of the stool as well as the wet weight of, which enabled us to identify the number of copies of the 16S ribosomal RNA per unit volume and per unit weight. We then carried out in the bottom row the total bacterial load calculation, which includes corrections for wet weight correction, collection or sample corrections for the dilution, and of course, copy number correction for the number of copies of 16S ribosomal RNAs that we would expect per organism in our samples. Taken together, what this enables us to do is measure the absolute abundance of microbes within a stool sample. So using this approach, we applied to samples that were either stored immediately at minus 80 without preservative, samples that were preserved in the OmniGene preservative, as is pictured in the purple, or samples that were preserved in the Zymo preservative, as is pictured in the green. You can see the temperature of intermittent storage or intermediate storage are arrayed on the x-axis. What you can see clearly here is that the OmniGene preservative actually led to an increase in the total microbes per gram that were measured compared to preservative. We actually think that this is, the OmniGene preservative does act as a little bit of a lytic agent, lysing some very difficult to lyse bacteria that evaded bead beating alone. By contrast, if you take a look at the Zymo preservative, we actually see that the concentration is lower than in the no preservative condition. It decreases with increasing temperature of intermediate storage, suggesting that here we may actually end up having some DNA degradation. Now, one of the things that we were interested to do was to understand how these preservative and storage temperatures might affect different phyla. If they affect all phyla the same, then our relative measurements should be similar between the different conditions, and it wouldn't really matter what condition we used if we were only observing these organisms in the relative space. So we decided to look at a ratio of organisms in the phylum Bacteria versus organisms in the phylum Firmicutes. For those of you who are not microbiome-focused researchers, these are the two most prevalent and abundant phyla in most human gut microbiomes. And the ratio of Bacteroidetes to Firmicutes has been suggested as a potential biomarker of various diseases. So here what you can see is the Bacteroidetes to Firmicutes on the y-axis, once again, the different storage and preservative conditions on the x-axis. And what you can clearly see is the B to F ratio is actually very different dependent on whether or not you add preservative. One of the things I actually find most striking is that while the OmniGene preservative is stable over the different temperatures, the Bacteroidetes to Firmicutes ratio is about 0.75 for the OmniGene preservative, as opposed to around zero point 0.4 for the no preservative condition. Suggesting that not only is the OmniGene preservative improving extraction of DNA, doing so probably preferentially for Bacteroidetes, such that we are seeing actually phylum-dependent effects on extraction of DNA and thus measurement of DNA based on these storage and preservative conditions. So taken together, I hope I've convinced you that it is possible to implement absolute abundance with a pretty simple to do 16S quantitative PCR. And we would argue that places where we think it's important to try and do this and where we and others collaborate trying to apply this is in figuring out, for example, whether absolute abundance might be related to whether or not people respond to immunotherapy or overall survival and cancer-specific survival in individuals with oncologic conditions. One of the things that we can ask ourselves, the absolute abundance of microbes in the microbiome be the most sensitive and specific marker of cancer outcome? I mean, the things that we need to look at. Taken together in this section of the presentation, I want to draw our attention to the conclusions that preservative and storage choices affect microbial composition. Real-world studies should carefully consider these choices for the reasons that I outlined before, and absolute abundance can and should be measured. In this work, we were able to revise the estimate of microbial to human cells from about 1:1 to 4:1. And if you're interested in more about this work, it was just published a couple of weeks ago in Biotechnology. So please feel free to take a look, read it. The protocol is well outlined there, and we're happy to answer any questions that you may have. So we've talked about absolute concentration of bacteria and viruses and how the microbiome. But one of the areas that we've overlooked is the viruses in the gut microbiome. Yishai Pinto and Meena Chakraborty, two very talented researchers in the lab, decided to team up to try and see if we could improve our lab's standard efforts to study bacteriophages and eukaryotic viruses in the microbiome, and therefore focused on developing methods to do virome-inclusive profiling. If we take a look at a comprehensive view of the gut microbiome, we know already that the microbiome is mainly comprised of bacteria. There are relatively few archaea and other eukaryotes. Most gut viruses are phages, which are viruses that, of course, infect bacteria. They're likely just as abundant as the gut bacteria. Despite this, historically, gut viruses have received less attention than gut bacteria. So why might this be the case? Once again, I would argue here that the reason this is the case is not because we don't think they matter, but rather our measurement approaches are highly biased against gut viruses. Most traditional methods to sequence stool samples only capture prokaryotic DNA using so-called 16S ribosomal RNA sequencing. This is a affordable, high throughput method that's well supported from an informatic perspective. And what it does is it actually amplifies a variable region of the 16S ribosomal RNA, which is part of the small subunit of the prokaryotic ribosome. Of course, only archaea and bacteria have the 16S ribosomal RNA, and so this, by definition, overlooks viruses and eukaryotes. A consequence of this is that the number of studies that have been published on the gut microbiome, as is pictured in the blue bars, far outpaces the number of studies that have been focused on the gut virome, which is in the red bars that you basically can't see here. Look at the number of publications by year, and as you can see, despite the fact that the number of studies of the gut virome is increasing, it's increasing by much, much less than the number of studies of the gut microbiome. Now, you may say, well, you can do shotgun metagenomic sequencing, and that should capture DNA from all gut microbes, including viruses that have a DNA phase. But probably are aware, standard computational methods to analyze shotgun sequence data do not accurately quantify gut virus. Now, this is because of the following. If you take a look at the picture on the bottom of this slide, on the far left, when we generate shotgun sequencing data, we generate both bacterial sequences in purple and viral sequences in yellow. However, when we map these to reference databases of genomes, we have many, many, many more reference genomes from bacteria in purple than we do from viruses. As a consequence, while we can relatively accurately quantify the bacteria, we do still a poor job of quantifying the viruses, despite the fact that these viruses are in our data to begin with. Because of this, researchers have needed an entirely separate assembly-based analysis to profile gut viruses. And very nice approaches such as VirSorter2, Vibrant, and others have been developed to essentially take all of the bacterial viral reads, assemble these short reads into contigs, as is pictured here, and to take these contigs and to do viral genome identification de novo, often using either homology-based or machine learning-based approaches. Once you do that, you can then quantify these viruses by mapping the raw reads back to the de novo genomes. So this work, unfortunately, it's resource intensive. It's limited in sensitivity to viruses that are abundant enough and well covered enough by your sequencing in order to generate an assembly. And of course, it takes extra effort. And as a consequence, in most standard microbiome papers, even if the authors generate shotgun sequencing data, they rarely go forward to the effort to actually do a based virome characterization. And that certainly was something that we had not done much in the past. Luckily, in 2021, several researchers started to pull together larger catalogs of gut viral genomes. This is just one such example, a manuscript that was led by Stephen Nafak and Nikos Kirpidis from the Joint Genome Institute. Here, what they did was they used metagenomic assemblies to create a compendium of almost 190,000 DNA viruses from the human microbiome. They titled the MGV catalog, and now that these databases exist, they're ready to incorporate into reference databases of genomes. So what Mina and Yishai did was they decided to introduce a called FANTA. It uses fast, accurate, reference-based approaches to simultaneously quantify gut bacteria and viruses. And it does so directly from standard shotgun metagenomes. If you're interested in this, we have a preprint on this. The pipeline is available on our GitHub. It is constantly being improved and updated. Use it. Please do share your feedback with us, and it'll soon be coming out in Nature Biotechnology. FANTA uses state-of-the-art tools such as Kraken and Bracken to quantify reads per sample against a virus-inclusive database of genomes. Basically, we created a comprehensive virus-inclusive database of genomes where we improve the representation of viruses by using this MGV catalog. First step is to classify using Kraken 2. Unfortunately, when you use these type of read-based classification approaches, you end up with a lot of false positives. And so one of the things that we needed to do in implementing our workflow was actually to filter out species with low genome coverage or false positives within the sample. We do this by eliminating organisms that have low genome coverage or a fraction of the genome that is covered by reads. We then quantify the abundance of each species in a sample by adjusting the read count per species for genome length and sequencing. And that allows us to then look at the relative taxonomic abundance as opposed to the read abundance of organisms within a given sample. Now, when we were developing this workflow, despite the fact that we were using already existing fantastic state of the art tools, a lot of what we needed to do was actually figure out how best to adjust parameters to make Fanta sensitive and specific. This entailed doing actually hundreds of simulations of gut genomes, as well as individual isolate simulations to assess both false positive and false negative rates. In our overall simulations of gut metagenomes, we included 200 viral genomes at a 5% read abundance and 500 prokaryotic genomes at about a 95% read abundance. When we carried this out, what we found was that Fanta classifies reads to the correct species and results in a 90% reduction in false positive species. Over here on the left graph, you can see that on a read basis, we have very high precision, recall, and F1 score. If you compare the default Kraken database to the Fanta workflow, you can see that on the y-axis here, we have many fewer false positive species called by Fanta as opposed to a default Kraken workflow. We then assessed the accuracy of Fanta in doing abundance estimates. And what you can see based on these simulations, here we have simulated relative read abundance on the x-axis and Fanta estimated relative abundance on the y-axis. You can see that most of our values actually fall nicely along the diagonal line, suggesting that our abundance estimates are accurate. We also included several post-processing pipelines or scripts that enable the user to determine relationships between gut viruses and bacteria within cross samples. When we apply this workflow to a data set of 245 shotgun metagenomes from healthy adults, this is from Yachida et al. From Nature Medicine in 2019, what you can see is that quantifies or measures five-fold more organisms or viral organisms in samples compared to the assembly-based approaches. Now, this is to be expected as assembly-based approaches require a relatively high abundance of those viruses and thus genome coverage in order to get an assembly. As expected, we see that Fanta is much more sensitive in detecting lower abundance organisms. When we take a look across these 245 individuals to identify prevalent phages I identify as those that are on the y-axis here. If we look at that by the mean relative taxonomic abundance of phages, you can focus on what's in the top right-hand corner. Those are organisms that are both highly abundant and highly prevalent. Those of you who are microbiome scientists are probably already thinking about phage, which has been described as one of the most prevalent family of phages in the gut microbiome of humans. But interestingly, we found that the most prevalent phages were actually hankyphage and love phage, two phage types that have been really described and that are quite prevalent and abundant. In conclusion, Fanta allows tandem profiling of gut viruses and bacteria in human stool samples. It is best suited for application on human gut microbiomes. And we are interested in trying to further develop this. Of course, that will depend on our field's ability to develop better catalogs of viromes from other niches, such as the skin, the vagina, and other ecological areas. Fanta is ready for further application to these human gut metagenomic data sets. We hope that what this will allow us to do is not only focus on the relationship between gut bacteria and human health, but also include gut viruses into that picture. We suspect that gut viruses not only impact health through altering composition of gut bacteria within the gut microbiome and changing the expression patterns of different antigens within gut bacteria and cell surface markers, but may also have direct impacts on human health. So with that, we've talked about absolute abundance and how important biomarker of human disease. We've talked about Fanta as a method to do viral profiling. And with the remainder of our time, I'm going to focus on a different topic, diving deep into the genomes of specific bacteria within the gut microbiome. Here, we'll focus on DNA inversions as a novel mechanism of genetic variation. So for this part of the presentation, we have to go way back to the year 1977. In 1977, a lot of interesting things happened. Elvis Presley passed away. Cher was born. And for those of you who are nerds, you'll know that split genes were discovered by Roberts and Sharp. Now, these split genes were the core of the finding of splicing of mRNAs and later on alternative splicing. And that was actually taken from their Nobel lecture. You can see that they compared higher organisms on the right to bacteria on the left, demonstrating that higher organisms had both exons and introns that could be then spliced to create different types of messenger RNAs and therefore different proteins. Unfortunately, bacteria do not have splicing for the most part. One gene encodes one protein. Now, this bacterial one gene to one protein model has stood strong since the time of the Bee Gees. So from 19, Bee Gees were popular, 22, when BTS was popular. The vast majority of the field, including us, thought that bacteria just have one gene, can make one RNA, and it can make one protein. Rachel Channon and Patrick West, two very talented postdoctoral fellows in the lab, decided to come together to try and think about this a little bit more and to ask, might there be heretofore undiscovered mechanisms of genetic variation in bacteria? Put another way, can we go from one gene to maybe two different messenger RNAs and therefore to two proteins? The core observation that was underlying this hypothesis was that DNA flipping is known to happen in bacterial genomes. This is an example of a capsular polysaccharide locus in a Bacteroides theta iota omicron organism. Bacteroides theta iota omicron, or B theta for short, is a common gut commensal, and it's often used in the field as a model gut commensal organism. In B theta, different capsular polysaccharide loci. And these capsular polysaccharides are exactly what the name sounds like. They are sugars that can coat the outside of the bacteria, and they're important for determining whether or not phages can infect that bacteria. They also act as antigens that our human immune systems recognize. Now, bacteria will turn on one capsular polysaccharide at a time, one of these 8. And the way several of these loci are controlled, turned on and off in B theta, are actually through the flipping of promoters that are upstream of these loci. There are invertases, which are enzymes that mediate DNA flipping, that can flip these regions and turn this locus on or off. Basically, these invertases flip around this piece of DNA that turns around that promoter. Just to review, this process is one of many processes that are called phase variation in bacteria. Regions of DNA, as pictured in the blue, are flanked by inverted DNA repeats, as pictured in the red, and these can be flipped in place reversibly. So it turns out there are lots of these intergenic invertons in gut microbes, especially in Bacteroidetes. In very nice work that was done by Zhang et al. In the groups of Allman, Xavier in 2019, what you can see is the identification of a large proportion of genomes within host-associated microbes that have these invertons in intergenic regions between genes. When you look at Bacteroidetes in particular, you can see that host gut-associated Bacteroidetes have around 90% of the organisms with invertons in these intergenic regions. What Rachel and Patrick then asked is, can we find uncharacterized invertible elements? The algorithm that Zhang et al. Had developed called FaceFinder and applied it to many stool samples that we had sequenced from patients undergoing hematopoietic cell transplantation. We focused on B theta alone with the idea that we could then follow up those findings with and more characterization, given that B theta is a genetically tractable model organism. And so we took these mixed metagenomic samples from about 400 individuals. We ran FaceFinder on these data and identified new inversions in the B theta metagenomic samples. We basically identified and figured out if these loci were in the forward orientation or in the reverse orientation. We focused on B theta because Bacteroides are highly abundant members of the microbiota that use this type of site-specific recombination. We know that expression of phase variable regions of these organisms, such as the capsular polysaccharides, influence microbe-microbe and microbe-host interactions. And of course, as I mentioned before, B theta is genetically tractable and it's a model organism for phylum Bacteroidetes. So any findings that we made, we follow up on. So when we were on these approximately 400 stool samples, we found over 350 invertons. Excitingly, though, we found some that were contained entirely within single genes, as is pictured in this yellow box. This was super surprising to us, as intragenic within gene inversions had never been described in the literature. And we didn't just find one or two, we found lots. By number 67 of these over 350 invertons were actually entirely intragenic. So what happens when you have an intragenic inverton? Here's a little cartoon. Here we have a gene in blue. We have the inverted repeats in the black. When you flip that region using a recombinase or invertase, what you end up with is a flipped region that's indicated in green. If that flipped region does not introduce a stop codon, we can end up with a recoding inversion, basically, which recodes the amino acid sequence of this green section. Alternatively, a premature stop codon can be introduced. That stop codon can either be followed by another start codon, meaning that this gene gets now split into two pieces, or there may not be a start codon, in which case you just have a truncated gene. These types of processes can untether domains, inhibit enzyme function, substrate specificity, alter enzyme kinetics, or create non-canonical functions. So we decided to dig deep and to really try and see if we could figure out what was going on. And to do so, we decided to focus on a set of examples. The first example we'll focus on is a prevalent inversion that we find in the Thi-C gene. Thi-C is a gene that's important for thiamine biosynthesis. Thiamine is a vitamin that is essential for de novo. Thi-C is a gene that's essential for de novo biosynthesis, and thiamine is, of course, an essential vitamin for B theta. Here you can see is that the inverted repeats or the intragenic inverton falls in between these two domains, and so probably will untether those domains. The inversion introduces a stop codon, so you end up with only this Thi-C associated domain, and you lose this second half protein. This validated in our metagenomic samples using both DNA-based approaches and RNA-based approaches. And then we went on to demonstrate that we can also detect this inversion in the B theta laboratory strain. So what are the consequences of inversion? To assess this, what we decided to do was generate locked invertons. So what's a locked inverton? Well, we took the B theta organism that was genetically manipulatable, and we basically obliterated this inverted repeat while maintaining the amino acid sequence. When people generate for intergenic invertons, usually they just mutate the inverted repeat. Doesn't matter what they do to it. They can just delete it. They can just mutagenize it because all they need to do is get rid of it to prevent this promoter from flipping around. Of course, when you're working in intragenic invertons, you can't just delete something or mutate it randomly. What you need to do is you need to mutate it in a thoughtful way, usually at the wobble position of the inverted repeats, so that you maintain the amino acid sequence. In so doing, we were able to generate locked invertons for both the forward and the reverse orientation. The forward orientation is the canonical version that leads to transcription and translation of the full gene. The reverse version is a version that enables the transcription of the full gene, but a premature stop codon for the translated product. We then measured the growth of these different B theta mutants, as is pictured on the y-axis. So this is maximum absorbance on the y-axis by a variety of different concentrations of thymine. As you can expect in the wild type organism, in the black line, the B grows just fine at all thymine concentrations because it can make its own thymine. Similarly, the locked forward orientation also can grow just fine because it can make thymine without a problem. If you take a look at the knockout, which is in the purple line, you can see that the knockout, as expected, of thymine can only grow when exogenous thymine is supplemented because it can't make its own. And as expected, the locked reverse mutant, which only has the N-terminal portion of the gene, so it has a truncated gene. That truncated gene is a loss of function mutant. And you can see that it has the same growth dynamics as the knockout. So you might ask yourself, well, why would a microbe encode a way to break thymine biosynthesis? Well, it turns out that making thymine is very energetically costly. This is a diagram on the right of the biochemical pathways that are involved in the production of thymine. As you can see, it's a complicated process. Thy-C is a very upstream gene in this process, and it costs a lot of energy for thymine. So perhaps in a population of bacteria, such as those that exist in the gut, only some of the organisms need to make the thymine, and then they can share that thymine with neighboring organisms. This might allow for more efficiency for the overall population. It may enable simple unicellular organisms to actually function as a population, and it may allow for specialization or division of labor within strains. And it suggests that there may be sharing of metabolites between close that are discordant for this inversion. So what are the consequences of this inversion? Well, we decided to use B theta Thy-C as a test case in the following microbiological experiment. These are pretty standard microbiology experiments, but as I know that we're a more computationally oriented audience, and indeed our lab was more computationally oriented, we relied very heavily on Rachel Channon's expertise as a microbiologist and the support of collaborators to help us think through how to do this experiment properly. What we did was we did a competition experiment where we took a forward organism. So this is a mutant that actually cannot flip the inverted repeat bound inverton reverse. And then we took a reverse version of the organism and we combined them. So we combined the ratio in defined media with thymine. We then grew them for 40 hours in our anaerobic chamber, and then we calculate the CFUs of the forward versus the reverse, and we figure out which one had a competitive advantage. Now, we did this at several different thymine concentrations, you can see on the x-axis, and we calculated the relative benefit that the forward had over the reverse. As expected, when no thymine is present, the forward has a great benefit over the reverse. The reverse, as you know, can't actually function well. When you take a look at concentrations of thymine that are actually common within the microbiome, you can actually see something interesting, where the reverse mutant actually has a little bit of a competitive benefit. suggesting that in situations where there is adequate or excess thiamine within the gut microbiome, B theta actually might benefit from turning off its thiamine biosynthesis operon through this reversion mechanism. Taken together, we conclude that at physically relevant thiamine concentrations, the Thi-C reverse is more fit than the Thi-C forward. So this is just one example of an inverton that we validated. We've also experimentally validated many in other invertons, including this integrase, BT0375. This is a really cool finding. If we come back to that CPS1 locus that I mentioned earlier on, where this promoter actually flips around thanks to this invertase, the inversion that we found in a really sort of meta way is actually within this invertase. When this flips, it actually introduces a missense mutation, which means that this recodes this CPS1 invertase. If we alpha fold the forward and the reverse orientation of this gene, you can see on the top sequence, the sequence in blue is the region in the forward direction. When you reverse that DNA sequence and you call the amino acids, you can see that the amino acid sequence. And as you can see in the blue versus the blue is the forward orientation and the red is the reverse orientation. This induces a structural change in the gene product. This structural change actually occurs in the DNA binding domain region, and we're in the process of autologously expressing and purifying this enzyme, trying to crystallize it and determine the actual structure, and to determine the functional consequences of this recoding inversion. So in the last couple of minutes, we're going to zoom out and ask, well, these intragenic invertons are interesting, but are they just a B theta thing? In which case, it might be very interesting to me, but it might not be as interesting to other people. To ask this question, Patrick West, who's a very talented computational biology postdoc in the lab, developed FAVA, which is an inverton finder that uses long reads. This works in a way that is relatively similar to phase finder, but unlike phase finder, which uses short reads as input, it uses long reads. We first predict inverted repeats. We then generate inverted and forward sequences in silico. So this is the forward sequence, this is the reverse sequence. We then take our long reads and we align them to both, and then identify inverted repeats with reads aligned in both directions. We can then quantify that are aligned to the forward versus the reverse, and we can determine within a population what proportion have the forward orientation and what proportion have the reverse orientation. To determine how wide this was across the tree of life, we downloaded about 30,000 genomes, long isolate genomes that are available in SRA. This was done with the help of colleagues from NCBI. These are representing about 4,000 different species of organisms. We then calculate the number of invertons, both intragenic invertons, as pictured with the blue dots, intergenic in green, and ones that partially overlapped a gene and partially did not in pink. On the y-axis, you can see the average number of invertons per genome. On the x-axis, I've arrayed various phyla. And what you can see is blue dots that are above 0 in many of these different phyla. Now, intragenic invertons are not incredibly common in isolates that are grown in rich media for long-read sequencing. But if we actually take a look at metagenomic data, I want to show you those data here today, we actually find the rate of intragenic invertons per genome is much higher in metagenomic long-read data than in isolate data. We also find lots of intergenic invertons, many of which we're now trying to characterize. We find these not only in bacterial phyla, but also as indicated with the asterisks in certain archaea. So in conclusion, we find that bacteria can have intragenic invertible elements that can lead one gene to result in different messenger RNAs that can result in 2 different proteins. I think this is pretty cool. So in conclusion, this is the first evidence to our knowledge of extensive within-gene intragenic inversions. We think that this is a new mechanism of gene variation. We've identified both truncating and recoding inversions discovered and validated, these inversions are widespread across the tree of life. Of course, there are many, many more questions that we're pursuing. We're trying to figure out what the biological consequences of these inversions are, which are regulated, what the ecological benefits of this might be for bacterial populations, and to ask the question, do these happen outside of prokaryotes, in eukaryotes? What about in things like cancer? So if you're interested in this topic, you can read our preprint, which is posted on bioRxiv. There's also a link to our GitHub repository for FAVA in that script, and we welcome you to use it and to reach out to us with questions. We think that these intragenic invertons have great potential applications, ranging from identifying new antibiotics from the same genetic clusters. We've actually found intragenic invertons in biosynthetic gene clusters, which might lead to different types of natural products being made from the same operon. We can think about ways to customize enzymes, both recombinases and methyltransferases, using these invertons. We can try to engineer programmable switches. Wonder if these might be underlying genes get presented on bacterial coats. And so it might help us better understand and control bacterial antigenic features to try and impact things like immunotherapy response. So in conclusion, about 3 different ways that we're trying to measure what matters in the microbiome. We talked about absolute abundance and also how preservative and storage choices make an impact on what we measure in the microbiome. I talked about Fanta, a new way to do virome-inclusive profiling. And I ended with a section that focused on DNA inversions as a mechanism of genetic variation within bacteria. And I highlighted the discovery of intragenic invertons. So with that, I'd like to thank our entire lab, our many collaborators, our funders, and of course, the patients and nurses who help participate in our study and enable us to study subjects. I highlighted the names of the individuals in the lab who were responsible for the projects that I shared today, and I'm happy to take questions.
Thank you very much for this really cool presentation. I found like they gave really a great overview over the relevant techniques that are really commonly used in the field and going to really complex methods. So I assume this will be really helpful also for people who are just starting to get a look at this whole topic and the microbiome field. So yeah, thank you very much. I was wondering what you said, like at the end on your first topic when you were speaking about these absolute abundances, that actually you found that the ratio of bacteria to human cells is 4:1 in the gut. Is that so? That's without any means of host depletion or without an infection present. I imagine that all of these things would impact those numbers.
Yeah, good question. So, these estimates rely very heavily on the estimates of prior researchers, including Sender et al. Basically, the one thing that we revised is, the number of human cells has been in the entire human body has been defined by other means or estimated by other means. What our approach did was we actually measured a much higher concentration of bacteria within stool samples than previous approaches had. And so that's actually what ended up modifying the relative ratio of bacteria to human cells. That the vast majority of bacteria in and on the human body exists in the colon. And so what our estimates did was really upgrade, as it were, the number of bacteria that we presume to be within the gut.
Oh, I see. Okay, thanks for that clarification. Yeah, that's kind of strange, isn't it? Like there's 4 times more bacteria there than actually there's human cells
It
in our
is.
body. So I was also wondering, so these mechanisms of how gut bacteria influence things like immunotherapy and cancer, what are the mechanisms, how that works? Is that understood?
Good question. There are some nice pieces of work that exist in the field and also are being developed to try and tease apart the mechanism. There are obviously many different bacteria that have been related to T regulatory cell proliferation, Th1 proliferation, Th17 proliferation. So Dan Littman has very nicely demonstrated that SFB, segmented filamentous bacteria, actually can lead to proliferation of Th17 cells. Work by Dennis Kapp demonstrated that polysaccharide A, which is one of the capsule polysaccharides that is present on Bacteroides fragilis, is actually sufficient to induce proliferation of T regulatory cells. Others like Kenya Honda have demonstrated that catabolic products of carbohydrate fermentation, such as butyrate, these short chain fatty acids, are also sufficient to induce T regulatory cell proliferation. We suspect that both metabolic products of bacterial fermentation and bacterial antigens that are presented on the cell surface of bacteria are probably important for and modulating immune cell function. We wonder if absolute concentration of bacterial products, such as different lipopolysaccharides, may also be important for tuning the immune system. And moving forward, one of the things that we and others are interested in is whether or not small peptides that are actually made by bacteria and secreted might be sufficient to signal to host cells, either through G protein coupled receptors or other receptors, to alter cellular function. And so I think moving forward, it'll be very exciting to understand how bacteria not only affect T cells and thus maybe immunotherapy response, but also how they might affect members of the innate immune system that communicate to the adaptive immune system, such as dendritic cells and macrophages.
Thank you. So that seems quite complex, all of it. And is it that these products kind of stimulate immune cells then locally in the gut area, or does it somehow traverse through the body where the T cells are maturing?
Good question. I think it depends. Yeah, I think it depends on the type of molecule. Short chain fatty acids are freely diffusible. And so assuming that some amount of those short chain fatty acids don't get used as an energy source by colonocytes, they actually can potentially get into the bloodstream and travel to regions of the body that may harbor more T cells. Alternatively, there may be breakdown of the gut barrier such that small proteins or even maybe big proteins or sugar antigens can actually traverse that barrier and make it to these sensor cells like dendritic cells that are actually surveilling the environment in the gut lamina propria. And so I think but both are possible, right? There are some molecules that actually can make it into the bloodstream. There are other molecules that can be experienced or seen by tissue resident dendritic cells, for example.
Okay. Well, thank you. Yes, it's quite complex. And I imagine, so how far are we from going somewhere really to these precision medicine approaches? Like, what would be an avenue to explore in relation to the gut?
Yeah, I think, one of the things I wonder about is if this kind of more careful measurement and attempts to try and measure not just organisms at the taxon level, but specific genes, gene products, and might enable us to identify highly reproducible alterations that are associated with response. So here's a hypothetical situation. Maybe what we'll find is that it's not the abundance or relative abundance of a bacterium that matters. It's the total abundance of Bacteroides lipopolysaccharides that's actually critical for determining immunotherapy response. Well, then we can think about how we might be able to modulate that by bringing up or bringing down the concentration of these Bacteroides lipopolysaccharides that the gut sees. Alternatively, maybe it's that there are certain antigens that are immunogenic within the gut. And these antigens may be surface antigens that can be turned on and off through DNA inversion or can be varied through DNA inversion within bacteria. Remember, it's in bacteria's best interest to actually find ways to evade the immune system. What if we can fix those in position, preventing bacteria from escaping immune attack? And this can essentially ramp up the immune system for things like improving immunotherapy response. And so those are some ideas of how, if we can really measure things more carefully at the level of the actual molecules that may have a mechanistic relationship to the host response to bacteria, we might be able to use that information to actually design therapies that will be smarter and more targeted than just giving people certain bacteria or bacterial cocktails back.
Yeah, thank you. Yeah, I think it makes a lot of sense, like this idea of absolute abundances. Like, I thought that was really a smart thing to look at. So I think we may be already at the end of the session. I'm not sure if we're still recording. Something said paused, but is there, yeah, there's some applause coming up. So I think the listeners are still there. Yeah, so thank you so much again, Amy. Amy, right? I
Yes,
want to
that's
say it.
correct. Yeah.
Okay, perfect. Yeah, some more applause there. So anybody still wants to give a question? Still a chance. All right. Well, then just thank you so much. I really enjoyed this, and I would be happy to ask more things also about the virome, which I found really exciting, about those catalogs. And so now, how would you, like, I imagine like one of the difficulties is to get at catalogs from these sites where the human cell load is much higher, I assume. Is that a major limitation, or you mentioned
Yeah,
that?
it is very difficult. Yeah, it's a good question. You know, the human samples are predominantly microbial, but let's say we want a catalog of a skin virome. That's much more difficult because if you do shotgun metagenomic sequencing of a skin sample, you're going to end up with mostly human sequence data. So there are ways to molecularly deplete human DNA, which might be a way to actually try and enrich viral samples. There are some classical methods to do viral particle enrichment through filtration. This, of course, selects for viruses that actually can form particles as opposed to those that are in a prophage state, meaning they're incorporated into the bacterial host genome. But I think we will need to think cleverly about how to create better catalogs of other viruses. There are other efforts that are being led to do marker gene-based identification of viruses that won't require probably such comprehensive catalogs. And I think that might be a really exciting alternative to the read-based classification approaches that we've described. And that may work better for certain niches that we can't generate as good catalogs for.